Hi everybody, welcome to Creation Myths. Today we are going to address the very persistent myth of genetic entropy in H1N1. The background here is that in 2012, Dr. Rob Carter and Dr. John Sanford published a paper called A New Look at an Old Virus, Patterns of Mutation Accumulation in the Human H1N1 Influenza Virus since 1918. In this paper, they claim the 1918 pandemic strain, or at least the lineage descended from the 1918 pandemic strain, went extinct in 2009 due to the accumulation of harmful mutations. Now, they don't actually use the words genetic entropy in this paper, but that's what they mean. And if you ask a creationist for an example of genetic entropy in nature, you're going to get a reference to this paper. Now, spoiler alert, Carter and Sanford did not adequately support this assertion at all. This paper is riddled with errors. In fact, I have in the past called this my favorite bad paper. There's just everything's wrong about this paper, and we're going to talk about it. So the argument goes like this. The 1918 strain of influenza accumulated mutations over the next 90 years or so. Over that time, fitness declined, and that lineage ultimately went extinct in 2009. This fitness decline and extinction was due to mutation accumulation, in other words, genetic entropy. Although again, they didn't use those words in the paper, but that's what they meant. So what you should know before we get into this is that fitness in the evolutionary sense, evolutionary fitness means reproductive success. It doesn't mean how fast or strong you are in the traditional sense. It's not about physical characteristics, it's about reproductive output. It's about, in effect, the representation of your alleles, your genes, in subsequent generations. Now, creationists like to redefine fitness into lots of other ways. For example, information content in a genome or structural biology, structural features. But these things obscure the actual definition, and they're all upstream of reproduction anyway. So like Dr. Sanford, in his book Genetic Entropy, talks a lot about information, and he says the genome accumulates mutations and loses information. Well, ultimately, for that to cause extinction, reproductive rate has to fall. So we're going to use the correct definition here, which is reproductive success. So problem number one with this paper is fitness of this virus. Carter and Sanford claim the fitness of this virus declined from 1918 to 2009. Now, the first issue with this claim is that they didn't directly measure viral fitness, and there are a number of ways to do this. And I was curious, because they didn't say anything directly about fitness in the paper, so I was curious, did they actually measure fitness in some way that they didn't mention? So I emailed Dr. Carter and I asked. I had a number of questions, but I said, and finally, did you compare metrics such as burst size, burst time, attachment rate, adsorption rate, or doubling time for the different genotypes across the H1N1 dataset? Basically, these are ways you can tell how fast a virus is replicating. The first few, burst size, burst time, attachment rate, and absorption rate, those are our components of how fast viruses replicate. And then doubling time is just an absolute measure. How long does it take that viral population to double? That's an absolute measure of fitness for viruses. Dr. Carter answered, no, we did not compute any growth metrics. Our work was done on computer only. So they didn't actually directly measure fitness for this virus, which, look, that's fine. It's influenza. It's a potentially dangerous virus. You don't necessarily want to be messing with it in the lab. That's potentially dangerous, and not everyone is set up to do that. That's a perfectly legitimate research decision. But that means if you're going to use proxies for fitness, you need to use the appropriate measures. As you might predict, they did not use the appropriate measures for fitness. The two measures that Carter and Sanford decided to go with for fitness are codon bias and virulence. And we're going to start with codon bias. Now, codon bias, this gets a little into the weeds, but in your DNA, when you have your genes, they're read, quote unquote, read 3 bases at a time, 3, then 3, then 3, in a non-overlapping code so it's just in sequence 3, 3, 3, 3. And those 3 bases tell your cells which amino acid to add to the protein that you're building based on a given gene. Now, those 3 bases for each amino acid, there's overlap in them. So, for example, CCC, CCA, CCT, and CCG all code for the same amino acid, proline. So you could have a CCC codon, or you could have a CCA codon, and those two different words say the same thing in the language. I'm using analogies here, but it's not actually a language, but in the language of the genetic code. Sometimes those codons are used approximately equally, but sometimes there's a big imbalance. Like 1 out of the 4 might be used 90% of the time, and the other 3 might only be present 10% of the time within a gene. When there's an imbalance like that, that's called codon bias. Now, viruses, you would expect them to want to match the biases of their host because they're dependent on the host for making their viral proteins. And that's a totally reasonable prediction. The problem is that influenza is an RNA virus, and RNA viruses mutate so fast, and codon bias has such a minute effect on fitness, it's basically undetectable, except in very extreme circumstances, which I'm not going to get in here. You put those two things together, fast mutation rate and very small fitness effects for codon bias, when you compare the virus to the host, and what you find is that variation in codon bias in RNA viruses is basically random relative to the host. And you can see here, this is an example from a 2013 paper where you have the host codon bias and you have the viral codon bias. These are RNA plant viruses, but it's RNA viruses like influenza. And you can see this is this is an almost random distribution. There's very, very weak association, if any at all, between the host and the virus. So there's no real correlation there. The second problem is that some of the changes that Carter and Sanford documented in the codon bias of influenza were actually adaptive. And specifically, there were changes away from codons that include the base C, cytosine, followed by G, guanine. This is important because the mammalian immune system recognizes CG dinucleotides. When you have a C immediately followed by a G, that is recognized by our immune system as something foreign, as an invader. Okay, so a virus will be under selective pressure in the human body to minimize the frequency of C followed by G, because that C followed by G is like a big spotlight that says, hey, I'm a virus. White blood cells, I'm over here. Come and eat me. Right? So you want to minimize that. And we do see those changes in the codon bias data that Carter and Sanford present in the paper. They ignore the fact that for influenza virus, those changes are actually going to be adaptive. The second problem, and really the bigger problem, is that they use virulence as a measure of fitness. Virulence is how sick a pathogen makes a person. Low virulence is something like the common cold, doesn't make you very sick at all. Ebola has extremely high virulence. It's an extremely severe illness. Now, optimal virulence depends on context. Basically, early in a pandemic, when nobody has gotten the virus and everyone's susceptible to it, there are lots of hosts around. So the limiting factor is going to be the degree to which an individual strain of the virus can compete within an infected host with other strains. Because if you make up most of the viruses within an individual, you're the one that's going to spread to other hosts. This leads to high competition within the host, and that's going to lead to selection for high virulence, because the viruses are going to win that competition by very rapidly infecting cells and making lots of copies of themselves as quickly as possible within that host. So it does a lot of damage to tissues. Now later in a pandemic, so that's the situation in 1918 when everybody is susceptible. Later in a pandemic, when lots of people have been infected, some people have even died, and a lot of people now have some degree of resistance to this virus, there are relatively few hosts available that are still susceptible. So the limiting factor in how a virus can spread through the population is actually between host competition. So the viruses in one host are competing with the viruses in a different individual to get to the few remaining susceptible individuals in that population. In that situation, you don't want to kill your host very fast, because even if you make up 100% of the viral load within an individual, if you kill that individual too fast, you're not going to find another new host before your host dies. So instead, the optimal strategy for the virus is to replicate more slowly, which means being less virulent, and that facilitates them spreading more widely through this limited host pool. So over time, we expect to see a decrease in fitness, and those changes are actually adaptive. They're beneficial. Now, this has been demonstrated experimentally in a 2007 paper by Heinemann and Bull. They were looking at bacteriophages, viruses that infect bacteria, and they basically compared phages in a high host density environment and a low host density environment, which mimics early and late in a pandemic. In the high host density environment, the virus was replicated extremely quickly, which means they would have had, if you kind of analogize it into humans, extremely high virulence.
In the low density host environment, the viruses were much more slow replicating, because if you're in a host cell, you don't have that many potential hosts around you, you don't necessarily want to just kill your host and move on, because you might not find another host. So the optimal strategy is to slow down and get as much out of the existing host as possible. This is exactly what we observe happen experimentally, and it's what happens in pandemics in the context of humans. So again, we have experimental evidence that those changes in virulence to H1N1, those are adaptive, which is contrary to what Carter and Sanford claim. Now, the second problem here is extinction. Carter and Sanford claim the pre-2009 strain of H1N1 went extinct. It did not. There was a new strain of H1N1 in 2009, and we'll talk about that in a minute. But Carter and Sanford actually accidentally acknowledged that the pre-2009 H1N1 did not go extinct. They wrote, similarly, there was a continuous and rapid accumulation of mutations over the entire history of the virus. And I want to highlight that phrase, entire history of the virus. And then they reference their own figures 2 and 3. Well, here's figure 2. And the circle here is in the original. That's not mine. But what they show here is mutation accumulation from, the red line indicates the 1918 sequence, and then they show the mutation accumulation over the 20th century into 2009, and then past it, and they actually include sequences up here that go all the way through 2012. Well, if you're going to compare mutation accumulation within a lineage of viruses from 1918 to 2012, which is what they do here, you cannot also claim that that lineage went extinct in 2009. You can't count mutations across that extinction event. So they implicitly acknowledge that the pre-2009 H1N1 lineage did not go extinct. They acknowledge it right there in black and white when they include the post-2009 sequences in their accumulation data on mutations. So they just directly contradict themselves right there in the paper. Now that's not all. There are other problems with this paper. So in relation to creationism, Sanford is best known for the 2005 book Genetic Entropy and the Mystery of the Genome, which I have a separate video about. I'll link that in the description. That argument in that book leans heavily on the concept of information in a genome, but it does not define it. Now, for H1N1, you would think if they're making an implicit genetic entropy argument, they would have addressed things like information, but they did not calculate the information content of the 1918 versus 2009 strains. I would have liked to have seen them calculate it and show that it declined, but they didn't do that. They also did not measure the activity nor the stability of any of the 1918 versus 2009 proteins. That would have been a useful way to document that the viruses were actually getting worse. And they did not calculate or compare the R0 of the 1918 versus 2009 strains, the R0 being the basic reproduction number for the virus. It's basically for every infected person, how many people do they give it to? And if you could show that the R0 was declining over time, then that might be at least pointing in the direction that the virus decreased in fitness over that interval. But they did none of these things. They didn't do any of these calculations, didn't show any of these data. And one more thing, and this one might be my favorite because it's just such a silly mistake. They calculate the mutation load over the 20th century by comparing a modern sequence to the 1918 strain. And we have the genome from the 1918 virus, so we know what that genome is. The sequence they use is H1N1 PDM reference strain A/California/04/2009 H1N1. And that's from a 2011 paper, and the emphasis there is mine. I want to make this very clear. The thing they're using to compare to the 1918 strain in terms of what mutations accumulated is an H1N1 PDM strain from 2009. PDM refers to the 2009 H1N1 swine flu pandemic. The problem here is that that is a completely different H1N1 lineage from the 1918 strain. And in fact, Carter and Sanford argue in this paper that the pre-2009 strain, the one that originated in 1918, was driven to extinction and replaced by the new H1N1 2009 swine flu pandemic strain. So we have two different lineages of H1N1 here, and in fact, we know we've done the phylogenetics on the 2009 pandemic strain, and there's a lot going on in this figure, but what I want to show you here is that the H1N1 from 2009 is derived not from human H1N1, but from human, right down here, H3N2. Okay, so it wasn't derived from the 1918 pandemic lineage, and then it became H1N1 through reassortment with other strains, avian and swine flu strains, and that's how it became a human form of H1N1. I'm sorry, you could probably hear a dog barking. So that H1N1 in the 2009 pandemic originated via reassortment. It is not simply a continuation of the 2009 strain, and that invalidates this whole paper. Their mutation count right off the bat is completely wrong. So to summarize, recall Carter and Sanford's argument. The 1918 strain of influenza accumulated mutations? I agree, that definitely happened. Fitness declined? They did not measure fitness. They did not use good proxies for fitness, so they have no way to say that fitness declined. The virus went extinct in 2009, according to Carter and Sanford. That did not actually happen. Their own numbers that they used in terms of the mutation accumulation timeframe shows that. And the above was due to genetic entropy. They didn't even try to establish a causal relationship between the mutation accumulation and the other effects that they saw. So the takeaway here is that the conclusions in this paper are completely unsupported. So, genetic entropy in H1N1, that is a creation myth. And now you know, and any time someone claims that an example of genetic entropy is H1N1 in the 20th century, thanks to Carter and Sanford 2012, you can tell them that is not the case, and here is why. So thank you for watching. If you enjoyed this, please subscribe, share, like, comment, whatever people do to get stuff visibility. Remember, always check creation sources and don't get fooled. Thanks for watching. See you soon.